Cell signaling is a complex and intricate process that allows cells to communicate and coordinate their activities. It plays a crucial role in various physiological processes such as development, homeostasis, and response to environmental stimuli. Understanding signal transduction mechanisms is essential for comprehending normal cellular functions and the dysregulation of these processes in diseases. In this script, we will delve into the fascinating world of cell signaling, exploring various signaling components, pathways, and their implications in human health and disease. Signal transduction is how cells respond to external signals and involves receptors binding to cells, triggering a series of events that lead to gene expression changes, protein synthesis, and cell behaviors. Cells have multiple ways of receiving and relaying signals to ensure accurate and specific responses. In summary, signal transduction pathways involve various components and terminologies. Receptors bind to ligands and initiate signaling, while adapter proteins connect receptors to intracellular molecules. Kinases add phosphate groups, phosphatases remove them, second messengers amplify signals, and transcription factors regulate gene expression in response to signaling. GPCRs, or G-protein-coupled receptors, are important for transmitting signals in cells. They interact with G-proteins, which are like molecular switches. This interaction leads to various cellular responses, such as changes in calcium levels or activation of protein kinases. Receptor tyrosine kinases are another essential class of cell surface receptors involved in signal transduction. RTKs are involved in various processes, such as cell growth, proliferation, differentiation, and survival. Unlike GPCRs, RTKs possess intrinsic kinase activity and phosphorylate tyrosine residues on target proteins. Upon ligand binding, RTKs dimerize, leading to transphosphorylation of specific tyrosine residues. These phosphorylated tyrosines serve as docking sites for downstream signaling molecules, such as adapter proteins, kinases, and regulatory proteins. The activated signaling pathways influence diverse cellular processes and are tightly regulated to maintain cellular homeostasis. These signaling cascades are critical for cell functions, and dysregulation of these pathways can lead to diseases like cancer. Understanding the intricate details of intracellular signaling pathways can greatly contribute to the development of targeted therapies and improving patient outcomes. Protein phosphorylation and dephosphorylation are critical regulatory mechanisms in cell signaling. Protein kinases mediate the addition of phosphate groups to specific amino acid residues, typically serine, threonine, or tyrosine. This activity alters protein conformations, interactions, enzymatic activities, and subcellular localization. On the other hand, protein phosphatases reverse these phosphorylation events, restoring proteins to their original state. The delicate balance between kinase and phosphatase activities ensures precise control of signaling pathways and prevents aberrant cellular responses. The cyclic AMP/CAMP signaling pathway is one of the most extensively studied second messenger signaling pathways. It is involved in diverse cellular processes, including metabolism, gene expression, and synaptic transmission. The main enzyme responsible for CAMP production is adenylate cyclase, which is activated by G-protein-coupled receptors or other intracellular signaling molecules. CAMP activates the serine/threonine protein kinase A, pKa, by releasing catalytic subunits from regulatory subunits. pKa then phosphorylates specific target proteins, modulating their activities and leading to cellular responses. Calcium ions, Ca2+, serve as crucial intracellular messengers in various signaling pathways. Intracellular Ca2+ levels are tightly regulated and change dynamically in response to extracellular signals. Ca2+ signals are involved in processes such as muscle contraction, neural transmission, gene expression, and apoptosis. Ca2+ signaling involves the influx of extracellular Ca2+ and the release of stored Ca2+ from intracellular compartments. Calcium-binding proteins, such as Ca-modulin, sense changes in Ca2+ concentration and modulate the activities of target proteins. Ca-modulin, for instance, can activate or inhibit enzymes, ion channels, and transcription factors. Small GT-PASAs are critical regulators of cellular signaling pathways. They belong to the Ras superfamily and include proteins such as Ras, RAC, and Rho. Small GT-PASAs function as molecular switches, cycling between active GTP-bound and inactive GDP-bound states. These GT-PASAs play diverse roles in processes such as cell growth, cytoskeletal dynamics, vesicle trafficking, and cell migration. They interact with specific effectors to modulate downstream signaling cascades and cellular responses. Nuclear receptors are a class of transcription factors that directly regulate gene expression. They are named so because they localize to the nucleus and bind to specific DNA sequences known as hormone response elements. Ligand binding induces conformational changes in nuclear receptors, allowing them to recruit co-activators or co-repressors to target gene promoters. The interaction of nuclear receptors with co-activators or co-repressors influences the recruitment of RNA polymerase and chromatin remodeling complexes, ultimately dictating gene transcription rates. Nuclear receptors play critical roles in processes such as metabolism, development, and immune responses. While G-protein coupled receptors are typically associated with G-protein activation, they can also initiate signaling pathways independently of G-proteins. These G-protein independent pathways involve the recruitment of adapter proteins, tyrosine kinases, or other intracellular signaling molecules. One well-known example is the beta-arrestin pathway, which can be initiated by several GPCRs. Beta-arrestins interact with the activated receptor and recruit intracellular signaling molecules, such as kinases or scaffold proteins. This pathway can lead to cellular responses distinct from those mediated by classical G-protein signaling. Dysregulated signal transduction pathways contribute to the pathogenesis of various diseases, including cancer, neurodegenerative disorders, and cardiovascular diseases. Mutations in signaling components or alterations in their activities can lead to abnormal cell proliferation, survival, and differentiation. Understanding the molecular mechanisms underlying aberrant signal transduction provides opportunities for developing targeted therapeutics. Many existing drugs and therapeutic strategies aim to modulate specific signaling pathways to restore normal cellular functions or disrupt abnormal cellular behaviors. In conclusion, cell signaling is a complex process that controls many cell functions. We examine the various components, terms, and pathways in signal transduction. From GPCRs to nuclear receptors, each part affects how cells respond. Studying signal transduction helps us understand diseases and find new treatment approaches. Further research in this field will lead to more precise and personalized medicine.